The main take-home message from 2 days ago was genes don't know what they're doing. Genes are recipes, they're readouts, that's all they are, and everything about what genes do has to be in the context of the environment regulating genes, turning some on, turning some off, and this paves the way for what's going to be an incredibly important concept starting next week and for the rest of the course. Different environments interact with genes in different ways. Gene-environment interactions, and that's going to be hugely important. Okay, so what we shift to today is two more pieces of this more molecular puzzle. Both challenge any sort of standard views in ways that are really pertinent to the evolution of behavior. Okay, first one, you get your genome. You get your genome from your parents. It's all in place by the time you're a fertilized egg, and that's it. You've got that genome forever after. And the first thing we'll be looking at is how the genome is not quite as stable as one might think. That there's more changes in it, in you, in individual cells over your lifetime, and with consequences of it. Now we start off with what was one of the most nutty pieces of biology history. Okay, so here we have DNA, and just in red, just this particular strand of DNA. And starting in the 1930s and 40s, a plant geneticist named Barbara McClintock started reporting something completely impossible, which was every now and then she was studying maize because it has some really clean phenotypes. It's easy to sort of figure out relatedness. If Mendel had ever heard of maize, he would have done the same thing. She's studying maize, and she was seeing some traits, some properties, some patterns of inheritance where she inferred the only way to explain what she was seeing was that every now and then a stretch of DNA is copied and then plunked down somewhere else in the genome. She reported this, and everybody said, oh my God, she's out of her mind. She used to be a decent scientist, but now she's gone crazy. And nobody paid attention to her. They paid attention enough to make up a term for what she was reporting, a totally snarky, dismissive term. People started referring to it as McClintock's crazy jumping genes. Ooh, every now and then our gene jumps, and nobody believed her. And as the legend goes, she worked in solitude and isolation. She worked out in the field with her maize plants at Cold Spring Harbor Lab, which is out in Long Island. She worked away because she knew what she was seeing, and she pushed ahead. And it took merely 40 years for molecular biology to be able to show she was absolutely right. Sometimes you will get stretches of DNA that are copied and plunked down somewhere else. And all of that snarky, jumping gene stuff went out the window, and instead they started to be called transposable genetic elements, or transposons. And everyone went crazy over McClintock at that point. She got her Nobel Prize. There were McClintock exercise videos. There was McClintock shoes, sneaker ads, all of that. Totally cool that she had worked in isolation all that time. And the coolest thing is that she was completely unruffled by any of it. When nobody listened to her for 40 years, she said, whatever, this is what I'm seeing. And when everyone suddenly said, wow, this is absolutely the case, here's a Nobel Prize, we're writing an opera about you, she also said, well, that's nice, but in any case, this is what I'm seeing. And she continued to work out in her remaze shortly before her death at 92, and totally cool, inspirational figure. Little bit of sort of debunking some of the majesty of that. In the 1940s, Barbara McClintock was already a member of the National Academy of Sciences, which is like the official old boys club of scientist secret handshake stuff. For her to have gotten in as a woman in the 1940s at a relatively young age, this is what they do for people when they turn 70 or something, she was a oh boy, she was completely established and highly respected, and people did pay attention to what she was seeing. It was cited, it was influential, but nowhere near as much as it should be. And what you're not supposed to say is part of it. The problem is that she was a very, very abstract writer where it was difficult to understand what she was. But in any case, she sees this, and by the 1980s, we have, whoa, every now and then, genes will jump. Ooh, I mean, every now and then, transposable genetic elements will do their transpositions. So the question, of course, becomes, what is this about? And what people began to realize was these stretches of DNA that were subject to these transpositional events. These were stretches that were derived from viruses, ancient viruses that had gotten into this species' genome and integrated into the DNA. And all this virus cared about was mechanisms to make more copies of itself, which gives rise, expanding on our concept of multi-level selection. This is not selfish genome or selfish gene. This is selfish DNA, serving no purpose whatsoever other than making more copies of itself. And in us, for example, about half of our genome, half of the DNA, are is descendants of these viral insertions, what are called retrotransposons, retroviruses, if you care about such things. So that's where the mechanism for this came in. These were these stretches that get plugged in by some virus way back when, and what it's good at is expressing the enzyme that will make a copy of itself and putting it in there, and thus and thus and thus, and that was the sole purpose of life. Okay, so that seemed to be the mechanism for what was happening. What are these transposons doing in any sort of way to make sense of it other than randomness? And in retrospect, it makes perfect sense that McClintock, as a plant geneticist, was the first one to find this. This was a real specialty of plants. How come? Because there's all sorts of circumstances where, something threatening happens. And what do? If you're an animal, you run away, you migrate, you move and plants can't run away when there is a stressful emergency. There's something happens, the environment becomes more challenging, who knows what. And what these transposable events were basically about was a Hail Mary. Okay, now that we have control over these things that copy them and plump them around, eventually they got taken over by the host cells. Now that let's just, make a copy of one random stretch of one of these and just throw it in someplace else and hope this will turn out to be something wonderful that saves our neck. The odds of that happening is you take a random 3-word fragment out of Harry Potter Volume 4 and plunk it down randomly somewhere else in the same book, and that shows you how to cure global warming. Like, this is desperation, but this is plants doing it during periods of stress where they just juggle around their DNA with these jumping gene things in the hopes of stumbling into something that will help them. So, great, McClintock, hooray for her, McClintock keto diets, all that, whatever, but okay, this is just plants. Yeah, we all respect plants, but there's her with her nice corn plants. And since then, what people learned is these transposons occur in all sorts of genomes. One of the most consequential realms is things that infect us, all sorts of parasites. There's this incredibly clever parasite called trichosamiasis, and you never want to get infected in it, but trichosamiasis, it comes and infects you, and what do? You got an immune system, so hooray for that. And what your immune system does is look at its surface protein, which is sure different from the surface protein on all of your cells. So the immune system, the alarms go off. This is something invasive, and let's start making antibodies that will attack this protein, that will recognize this protein and thus make it possible to go in and destroy this parasite. So let's do that, and that takes, I don't know, a week or two to build up enough antibodies. And what did this damn pathogen evolve as a capacity? About a week into it, it juggles a whole bunch of its genes and comes up with a new surface protein. Just when the immune system was ready to recognize the old one and attack it, it's gone invisible. It can't be seen by the immune system anymore. And, you got to go and make a new antibody. And a week later, just as you're about to get there, it does it again. It does the same thing. And this is an extremely effective means by which a pathogen can evade a host immune system. So, what do you expect to see at that point? The immune system evolved to do the exact same thing. That was that major histocompatibility complex thing from last week. This is a means by which the immune system, gearing up to make an antibody against this novel thing, gearing up consists of juggling a whole bunch of genes in this major histocompatibility complex and seeing, okay, we just made 300,000 new novel antibodies, and let's see if any of them happen to react against this thing, and then get rid of the rest of them. So all this transposon stuff is highly exciting and relevant to all of us. And of course, what's really relevant to us is to now ask the question, so does this stuff happen in the brain? And the answer is yes, under all sorts of really interesting circumstances. This was mostly the work of someone named Fred Gage at the Salk Institute, major figure in the field. And what began to be figured out was, first off, so what's the enzyme that does this copying? And people had to come up with a name for it. And in a moment of extreme non-creativity, they were called transposases. These were the enzymes that does this. But what Gage started to explore was, does this happen in the brain and when? And the first thing he found was totally cool. Just when a stem cell is turning into a neuron, there's an explosion of transpositional events. What does that mean? Right around the time you were making a new neuron, you decide this is a good time to just get some variability. And with this one, let's juggle some of our DNA and see if anything cool arises as a result. What's the implication of that? The genomes of every single one of your neurons in your brain not only differ from what you got from your parents, but they're likely to differ from each other. You have a whole mosaic of different genomes and different neurons. So that happens right when you're making a new neuron. What was next shown, again, his group showed this utterly cool thing, that different mothering styles in rats, back to all of that, different mothering styles stimulate different patterns of transpositional events in neurons in the brain. Whoa, totally cool as well. And then what you see is when you are stressed like not plant stress, there's like no rain or whatever, but stressed in all our everyday sense of it, stress hormones are capable of triggering some transposons to jump around, inhibiting other ones from doing that. And so this is all happening in the brain. What's the conclusion? Your genome you got from your parents not only is different than what you have in your brain, it is different in every single neuron, and people are just beginning to explore the functional consequences of that. Now, you get another route by which you can get variability in your genome, where the thing you got from your parents way back when is now different in different cells. When a cell divides and undergoes mitosis, and now there's two cells, and part of what you're doing is copying the DNA, there's copy errors that sneak in. So you get a certain amount of mosaicism in the cells there. And here's an example of mosaicism in the brain, which nobody knows yet if it really matters much. And I'd be surprised if it does, but it is so cool. It is called Microchimerism. Okay, actually, it's got two names. It's either called feto-maternal microchimerism or it's called materno-fetal microchimerism. What is this about? When you are pregnant, some of your fetus's cells kind of slough off and somehow get into your circulation. And when you were a fetus, some of mom's cells up there have sloughed off and gotten into the circulation. And there's a certain number of stem cells that get into the mother from the fetus, and a certain number of maternal cells that get into the fetus and get incorporated. Whoa, that's interesting. And do some of these cells get into the brain? Yes, this has now been shown, including the human brain. Are there a lot of them? No, not really. How do you tell if there's a neuron sitting in there that, was originally a stem cell from your fetus and now it's in your brain? You look for a really traumatic version of that. You were pregnant with a son. And every single cell in your body, every neuron is XX. And then they look, and every now and then you see a neuron that's XY. Whoa, that's a cell that came from the fetus, my son, the fetus, and got in there and presumably is all over the rest of the body. That's the most dramatic version of it. And you wind up then looking in the nervous system of sons of mothers where this was going on, and you will see amid an entire brain of XYs, there'll be some XX neurons. Whoa, those were once part of mom as stem cells, and so there's this chimeras, these sort of mixtures of different origins of cells in there, including neurons. Okay, that is immensely cool. It's not at all clear how important it is, but it's just like irresistible that that happens. And what is also seen is the more pregnancies someone or some rat or something has had, the more of those, the larger the number of neurons, because each time this transfer goes on between mother and fetus. What's the punchline here from what we've just seen so far is through a number of different routes, this chimerism, mitotic copy errors, and transposable events. The genome you got from your parents when you were a single fertilized cell is not what you're going to have as an adult. It's not that stable, with interesting consequences. Now we switch to what is really the most important topic here. After we get under our belt the notion that genes don't know what they're doing, now let's look at how mutations, boring mutations, point insertion deletion, let's see how you can have types of mutations that cause really dramatic radical change in cells, in you, in the genome, in all of that. So what was the backdrop for people thinking about this? And here you had to go hang out with paleontologists, which was a very odd thing for molecular biologists to do. Paleontologists, and what you would see is you'd get some extremely complete fossil records of something or other. You'd have a gazillion fossils evenly spaced over time, and what everybody expected to see is gradualism. You would see over time some characteristic of this fossil changed over these millions of years or whatever. And instead, what paleontologists were often seeing were long periods where nothing changed. Long periods of stasis, of equilibrium, long periods where seemingly selection is not really going on in all that interesting of a way. And what would also then be seen is, in the blink of an eye, you'd get a dramatic transition. Rather than nice gradualism, you would get some revolutionary jump in some property of some fossil. Interestingly, this was first introduced as an idea by Stephen Jay Gould and Niles Eldredge. Stephen Jay Gould, famous Harvard paleontologist, all of that, who also happened to be a very committed Marxist. And it was kind of cool. He was not hesitant to put this in the context of, no, this is not reform, this is irresolvable dialectical forces, which when they resolve, jump dramatically. And this was this notion that often the fossil record looks like this, or would look like this, and suddenly this one appears as well. There's been speciation, or like this species went extinct but split off in two other branches, and that was totally cool. And like in his view, what the stasis meant was the last 2 weeks worth of behavior stuff. Ooh, again, you want to be 1.5% more effective at being aggressive at the right time to leave more copies in your genes. Most of the time, nothing is happening. Most of the time, that bloody and tooth and claw has no consequences. And that getting good at playing the prisoner's dilemma has no consequences either. So, all sorts of people were not amused by this and attacked this whole argument along a number of lines. First as a rebuttal, these are paleontologists saying, whoa, look at these changes in a blink of an eye, this jump here, this very incredibly fast. You're a paleontologist. What counts as fast? 100,000 years or something like that. So you're supposed to like make neurobiologists impressed that behavior evolves in big jumps like this, and say 100,000 years. We can become like, we could invent poetry during that time. Who knows what counts as rapid for a paleontologist is not what counts as rapid for this sort of world or for neurobiology. Here was the next response, which was looking at this and saying, well, what do these paleontologists look at? They look at paleontological stuff. They look at fossils. Gould was like an expert on the evolution of snails or something. You're looking at things that are preserved as fossils. Shape, morphology, shape of the snail shell changing in this way or that way over time. And you're sitting there and he's saying, oh, the whole emphasis of competition and cooperation, all of that is meaningless because most of the time there's stasis and there's no. And you say, are you kidding me? You were making a global pronouncement like that based on what you do, which is looking at the shape of snail shells? That's telling us anything interesting? Every time you come up with a new type of thought, a new type of neuron, your skull doesn't suddenly change shape. By definition, what paleontologists were looking at with their fossils was stuff that's like just body shaped and bones and boring as hell, and they couldn't say anything about what was going on inside. And here's a paper called Fossilized Nuclei and Chromosomes Reveal 180 Million Years of Genomic Stasis in Royal Ferns, deciding that they could understand what's happening by like just looking at like the shape of cells or things like that. And this just struck me as ridiculous. I have no idea why I sound so bitter about this, but in any case I am. I'm outraged that royal fern people would let this slip by. And the other rebuttal was, what's the mechanism? Show me a molecular mechanism by which change can happen this quickly, which is what the rest of this lecture is about. So, a couple of different mechanisms by which you could have very rapid evolutionary change. So, one way, thinking of transposons here, is this is just some random junk DNA, and you've made a copy of it, and now you got 2 bits of random junk DNA. Every now and then, what you see instead is somewhere during DNA replication, something like that, a mistake happens, and now this red thing is a gene, and you make an extra copy of the gene. And what you have shown here is gene duplication. When it's duplicated, it's very often right next to the original one, an extra copy there. You will see duplication, multiple routes of duplication of many genes. Whoa, what's that about? All sorts of interesting things that happen when you have gene duplication. One example, you now could have twice the amount of gene transcription to make twice the amount of protein if you feel like it. That's good. Next thing is you have a backup gene in case something happens here. Okay, that sounds ridiculous, but this was actually very relevant to Alzheimer's disease research a while back, in that there's a gene that's been very heavily implicated. People who care, it's the APP gene for amyloid precursor protein. Some of the best evidence for it was finding some families with a mutation in this gene, and all of them got Alzheimer's when they were like 50 years old. Incredibly interesting. And thus, if you really want to prove to the world that this gene matters, let's have some mouse versions of this, and let's engineer the mouse to be lacking this gene. And if this gene has anything to do with Alzheimer's now, they never get anything like Alzheimer's. What does mouse Alzheimer's disease look like? It is a very modeled version of it, but we could ignore that. Whoa, if this is how the world works, you should never see mice getting mouse Alzheimer's. But instead, huge disappointment in the field. It had no effect at all. They still got mouse Alzheimer's disease, and this was finally resolved when they realized, oh, there had been a duplication of this gene. And when we knocked out this one with genetic engineering, this is the one that took over the role. So sometimes what you see there is it's a backup. It's sort of an alternative there that you've got available. What you mostly have here is this great evolutionary freedom, because now you've got your original gene that's presumably there because it's doing something useful, and you have this backup, this duplicate, in a silenced mode, and you allow it to just drift. It's not subject to selection because it's silenced, and little changes come in with it over time, and just like that, little changes come in, and you're just letting it drift randomly. And every now and then you see, have we come up with anything interesting here? And most of the time it's gibberish, but every now and then you've just invented a new type of protein that's coded for by this. And a great example is there's a whole sort of evolutionary history of receptors for different steroid hormones, and one can see that these two similar hormones had a single ancestor at one point and produced a hormone that was a mixture of the two, and there was some gene duplication event, and suddenly you had two separate ones. In the same way, go back 100 million years, and these two hormones, oxytocin and vasopressin, that we have already heard about, was just one hormone with one gene called vasopressin, and at some point there's a gene duplication, and you got to invent a second hormone out of this while this drifted, and you held on to that, and you get rapid change that way, dramatic change. So, gene duplication, that is one possible mechanism for things happening rapidly. And when we eventually get to the lecture on schizophrenia, you'll see there's some evidence that abnormal numbers of copies of certain genes or stretches of DNA may have something to do with it. The most important realm in which we see how you can get dramatic changes in genes and their products is now showing something that I lied to you about the other day. Okay, what did we go through? We went through this major revelation that DNA is not structured. So here's one gene, and here's the next one, and here's the next one. And instead, we've got 98% of DNA is non-coding, and that's the instruction manual and promoters and suppressors and all of that cool stuff, regulating a gene which looks like this in terms of its DNA, this continuous stretch of DNA. And in the 80s, major revolution, mostly this guy at Harvard named Wally Gilbert, began to notice as sort of gene sequencing techniques, DNA sequencing techniques got better, seeing, whoa, this protein, which is obviously coded for by this gene, when we really look, we see this gene is broken into 3 separate pieces. Whoa, that's bizarre. And then stretches of non-coding DNA in between. This is completely bizarre that it turns out most genes in us, in mammals, etc., instead of it being coded for by a single continuous sequence of DNA, is broken into 3 separate parts, 400 separate parts in some cases, massive numbers of these. So people had to come up with a term for this, and Gilbert was the one who said, okay, here's what we're calling the parts that code when put together for a particular protein. We're going to call those exons. And the intervening part that don't transcribe, these intervening parts, we're going to call introns. Exons and introns. And this was hugely revolutionary because it turns out most genes have actually an exonic intronic structure to them. So, first question is, so how do you get a single protein out of this? And what you then see is you make a stretch of RNA that covers all of this, both the exon, intron, and then was discovered a class of enzymes called splicing enzymes. And what one particular specialized one would do is come along and splice this part out and splice this out, and then stitch these 3 together, and you now can code for the complete protein. Totally cool, totally amazing, totally like, what? Where? Why do things this way? Why do things like that? And I don't know why it happened in that teleological sense, but what you begin to see is, whoa, does this open up all sorts of possibilities? Because there's different types of splicing enzymes, which splice in different places. And suddenly, you see in some circumstances, you get all of this is spliced out, all of this stuff is destroyed, and you only get protein A1 as a result. Meanwhile, a different splicing enzyme is junking all this and junking it, and you get protein A2, a completely different protein, right? And this whole combinatorial thing of 1, 2, 1, 3, 2, 3, 1, 2, 3, all of that, with a whole array of these splicing enzymes. Look at what you were doing here. One gene, because it has 3 exons, can generate 2 to the third minus 1, can generate 7 different types of proteins. And this blew everyone away because, for one thing, like, ooh, does this count as 1 gene? Does this count as 7 genes? Which is why when you look up, so how many genes do we have? And they always say something like 20,000, because there's still something of a debate in a circumstance like this. Should this count as 1 gene, 7 of them, all of that? And 75% of our genes undergo splicing like this, where you could make multiple different outcomes. Wow. Totally amazing in that regard. Totally amazing in that it takes us back to our theme from the other day. DNA sequence is kind of cool, and changes in that are certainly interesting if you get a change here or there. But the regulation of it, the splicing enzymes, which splicing enzyme gets activated, is going to determine which protein is made from this. And that can have, obviously, enormous consequences in opening up a whole universe of what regulates the splicing enzymes. And, we are often running enzymes are proteins. Therefore, there's a gene for splicing enzyme 1 and a gene for splicing enzyme 2. So environment might have turned on splicing enzyme 2. And as a result, there's lots of this enzyme around, which does cleavage producing this one. And in another environmental circumstance, it's this one that's made. And we've just got this whole new level of regulation of things derived through genes that regulate how other genes are processed, with environment influencing the genes that give rise to this, and the genes that start off there, and layers of regulation like this coming in. But the term in the field is there's all sorts of possibilities of alternative splicing, cutting and pasting in different ways that are combinatorial and generating 7 different protein messengers out of the same gene. And which ones you generate depends on which ones have been activated by environmental cues. And you will see things like there's this protein, there's this gene called POMC, pro-opioid melanocortin, and in the anterior pituitary, splicing enzymes process it in a certain way as to produce 3 different hormones. And in the posterior pituitary, different alternative splicing enzymes tackle it in different ways and come in different places there and produce 2 other hormones completely separate. That we've got this enormous capacity for variability coming in here. And what we begin to see now, just to make things more complicated, is you can have promoters in the introns, in the intrav. You could have different promoters. Not only are we like having splicing enzymes that can cut and paste different ways, you can even have promoters that will mean that only A2 and A3 is transcribed. So here we are. These are 3 different genes, A, B, and C. And you know by now, A is probably not a continuous stretch of DNA. It's probably broken into exons, that kind of thing. But we have A, B, and C. And we've got C is under the control of this promoter binding this transcription factor. B is under the control of this transcription factor. Oh, that's supposed to be complementary, so this has a little fin sticking out. Let's make sure you got the shapes right here. So that's what this transcription factor looks like, and it plugs into there. And then in gene A has both promoters. Gene A could be turned on by either transcription factor 1 or transcription factor 2, multiple promoters at the same gene. And what's interesting about this, these 2 different transcription factors may turn on gene transcription for different lengths of time with different intensity, all of that, coding for different sorts of versions of the protein being made. What does that take us back to? From about 20 minutes ago, our gene environment interaction stuff. What do we mean by this? One environmental circumstance leads to activation of transcription factor 1. Another environment does this, and they both cause more copies of this gene to be transcribed. They cause more of the protein to appear. But what you see is, because of the differences, 2 different environments will produce different amounts of a protein, or maybe even mildly different versions of it. Multiple promoters is a basis for gene-environment interaction. Activating a gene is not always the same. Different sorts of environments activate the same gene in different sorts of ways. And we will see all sorts of incredibly interesting stuff. Just as one bit of dramatic foreshadowing, we're going to see a gene that turns out to be highly relevant to major depression. And it is under the control, like big surprise, of multiple promoters. And what you see is only one of the transcription factors actually activates transcription, and this is activated by stress, producing a protein that winds up being relevant to depression. So we've got this first enormous amplification of our whole story here. You got multiple different promoters upstream of a particular gene. You've got different routes by which environment can turn it on. So that's immensely cool. And what people have seen by now, I'm forgetting what the record is, but I thought I saw a paper some years back where people had identified some gene that had more than 100 different types of promoters upstream. 100 different types of environmental circumstances would activate this gene to different degrees with a different phenotype as a result. Whoa, we've just gotten so much complexity out of this. Notice embedded in this diagram is a whole new level of complexity, which is this particular promoter is found on gene A and gene C. This promoter is found on A and B, which means that when this one is activated, you're going to produce proteins A and B. When this one is activated, you produce A and C. What have we just jumped to? A critical word here, relevant in all of this. The word network has just come in. One transcription factor will find promoters, lots of different genes will have the same promoter, and thus a transcription factor doesn't just lead to the transcription of one gene, it will lead to a transcription of a whole network of genes. A different transcription factor, which could activate the same gene, is at the same time activating a completely different network, overlapping networks of things being activated, which is really, really interesting because we're now moving into a realm of rather than what one gene does, what does a whole network of genes do? And there's a great example of this in people who study sort of inflammation. There's a transcription factor called NF-kappa B, which is activated by all sorts of different types of cell injuries. And there's a scat of different genes with upstream promoters responding to NF-kappa B. And what happens when it's activated, it turns on expression of like a dozen different genes, 10 dozen, who knows what. If you go away for a weekend and look at the journals on Monday, there's another gene that turns out to be regulated. And what do all of these genes, protein products, collectively do? That's what causes inflammation. The inflammatory response is mediated by this one transcription factor, and you get an entire network of proteins that bring about inflammation because you have an entire network of the same promoter being upstream of a number of different genes. You're getting massive, potentially rapid change by making a copy of a gene, and who knows where it will drift into after a while, but you can make double the amount of protein. But even more fundamentally, you can have different environmental circumstances activate the same gene, but with subtle differences in how they do it, or different environmental circumstances activating completely different networks of genes. Massive amplification here of, ooh, so what happens when you activate this transcription factor? Enormous consequences versus when you do this one. This is where you've got all sorts of networks that are partially overlapping. Whoa. So what happens if you have a mutation in any of these players here? Any of these, a mutation in an exon, a mutation in a promoter, a mutation in the gene for a transcription factor, and each one of these is potentially going to amplify with enormously broad consequences. So you get a mutation in one given exon there, and potentially you are now making a novel version of 4 of these different proteins. What is it? This one by itself. 1, 2, 3, 4 different proteins. And whoa, one mutation, and now you've changed the function of 4 different proteins. And this is the other days like boring textbook mutations, point, deletion, insertion. Doesn't have to be a fancy mutation, but being on one particular exon, it's going to affect a subset of the proteins that are under the control of this gene. So get a mutation and suddenly in the exons, and you've got a dramatically different picture of what the protein products are. Things can change dramatically because a whole bunch of proteins are working differently rather than the other day's gradualist picture of, okay, so this gene now makes for a protein that does a little bit more of this or a little bit less than that. And here we see one of Wednesday's classic little textbook mutations, amplifying and the consequences of this. Second realm of mutations. Now you get a mutation in a promoter so that different environmental circumstances turn it on. This protein's product, this gene, is now part of a completely different network than it was part of before. Whoa, that's majorly amplifying. And down the line, we're going to look at two species of voles. We're going to look in great detail on them. And one species is the mountain vole, and one species is the prairie vole. And they're really closely related. And you'd expect them to have fairly similar social systems, but no. One of them is a pair bonding species, one is a tournament species. And people began to figure out the difference, and the difference was driven by the males. Do they stick around after they mate or not? Or do they head for the hills and are never seen again? It's got to do with the hormone vasopressin and what it's doing in the brains of male prairie voles versus mountain voles. So everybody gets all excited and saying, let's just get the sequence of the vasopressin gene, and I bet you it's going to be different in prairie voles and mountain voles. And it was identical, the same exact gene. Whoa, that's sure confusing. What's happening here? And eventually people figured out what was the difference in the DNA sequences relevant, not the vasopressin gene, the gene for the vasopressin receptor. Aha, okay, so that's the thing where mountain voles and prairie voles have a difference in the promoter, a minute difference, unless you get something very interesting there. When it binds vasopressin, it activates the neurons there. Where are those neurons? In the dopamine reward system. Aha, what does that help to explain then? You know, you're a polygamous male vole and you got the usual system, and that includes having no vasopressin receptors in this reward area. So you mate and you secrete vasopressin for whatever reasons, and it does its thing, and then you are on your way and never heard from again. But if you have had a mutation so that the location where this protein is expressed, and suddenly you've got this receptor in the reward pathways in the brain, the male mates, and it's super duper rewarding. So he sticks around and does it again and does it again, and you've just invented a version of pair bonding. You've just invented mating is more pleasurable for the pair bonding males than the polygamous ones. How come? Because the pair bonding one has vasopressin receptors in the reward area part of the brain. Where'd that difference come from? Not because the gene differs, but because of a difference in the promoter, a difference in the networks of expression in this part of the brain versus that part of the brain. And we will hear eventually about one of the all-time cool studies where they did some molecular fumfering with the genes in these voles, and they stuck a pair bonding vasopressin receptor gene promoter into one of the polygamous vole species, subspecies, and what you did was you converted them from being polygamous to monogamous. And that, of course, immediately raises the question of should this be labeled as gene therapy, of a way of like curing polygamy, or but in any case, that was totally a single mutation producing a different type of promoter, and you get two species with totally different mating systems, and thus you get what turned out to be two different species. You've just had a major jump like that. So we see huge consequences when there's a mutation in one particular exon. Magnified, amplified, amplifies our word over and over here. When you get a mutation in a promoter, you're going to have amplified effects. It's now going to be part of a different network. Or what about that like fact from the other day? Transcription factors are proteins, so there's transcription factor genes. So what if there's a mutation in the transcription factor gene or in one of the exons or in one of its promoters, and this transcription factor is going to be a different one now. And the genes under its control are going to be part of a completely different network. So this amplifies also. And we have a fourth domain of this, which is the splicing enzymes. They are proteins, and thus there are splicing enzyme genes for all the different possible types, and you get a mutation in the splicing enzyme number 1 gene or its promoter, and now you've got completely different clipping and alternative stitching with pieces together. What we've got here is enormous capacity for expanding minute little classical textbook little piggly point mutation or insertion or deletion. You could just delete one base pair, and that's this tiny little thing which amplifies like crazy and changes entire networks of gene expression. And as we just saw with the voles, this could be consequential enough as to cause speciation, because mountain voles and prairie voles have almost identical genomes and just differ here or there in a few spots, like the promoter for the vasopressin receptor gene. Enormous consequences. And we've already seen one version of evidence for this, noted the other day. When you look at, some chimps, we have 98.5% of our DNA in common, identical, all of that. So what are the differences? And when you look at the genes that are different between humans and chimps, a disproportionate percentage of them are coding for transcription factors. In other words, little tiny differences between us and chimps, only involving like 1.5% of our DNA, have big amplifying consequences. The ability to make new networks. So what might this look like now on our evolutionary level? Because what we're now introducing, when you change a network, you change an if-then clause. If and only if this is happening in the environment, I activate this transcription factor, and as a result, these proteins are made from this consequence. And potentially, thanks to one of those amplifying classical mutations, and one of the key spots there, you amplify, you create new if-then clauses. Let's see some examples of this. And a lot of these are built around some hormone effects. Okay, steroid hormones. Do not freak out at this if you've got no endocrine background. The catch-up section won't get you up to speed. This is just like an example, but a very cool one. So, steroid hormones, estrogen, progesterone, testosterone, glucocorticoids, aldosterone, a whole family of these with histories of gene duplications. And that's how you started getting different pathways. So you got a whole bunch of different steroid hormones, and they're structurally very similar, and this whole family, and you better bet you got to be very, very subtle there with how you deal with these structurally similar molecules, because if one of them is estrogen and one of them is testosterone, you do not want to confuse the two. You want to be very careful about distinguishing them. So you now look at the receptors for steroid hormones, and when you see their construction, this was an intensely hot topic in the 90s, there's a, quote, modular construction to steroid hormone receptors. What do I mean by modular? Every given steroid hormone receptor has two parts to it. You've got the hormone binding domain. And what happens when a steroid hormone receptor binds its hormone? It changes shape and becomes a transcription factor. It changes shape it reveals a binding site that plugs into a promoter. These hormones have their effects by working on receptors, which, if and only if they are occupied, have a conformational change and now become a transcription factor that plugs in here and controls transcriptional events. So what is modular about this receptor? There's the hormone binding domain and the DNA binding domain. And that's a very, very exonic gene that codes for that, and a whole bunch of separate exons coding typically for this part of this molecule, and others coding for that part. There's different modules, and they're coding for hormone binding domain and DNA binding domain. So you can now see what this introduces is the possibility of our amplifying mutations. So suppose you've got a gene-environment interaction network, and it goes as follows. You are some desert rodent or some such thing, and you've got a whole network which, when it's really dry out or during the dry season, then the dry season, there's not much water and it's not raining, and you've got all sorts of means by which your body senses this. And what happens is, down in your kidneys, when it's dry, when there's not much water, this activates a transcription factor, which does something or other to your kidneys, and you retain more water. And hooray, you are less likely to desiccate now as a desert rat. That's terrific. Let's suppose there's a mutation so that what used to be the hormone binding site for one hormone now gets coupled to the module of a DNA binding site for another. We have a different circumstance that will activate the same gene, or we have the same circumstance activating different genes. And in this case, suppose things are juggled with all of our mechanisms here for big amplify. And now you've got one where when it's dry out, that environmental signal, among other things, also causes you to ovulate. What have you just invented? What have you've invented seasonal mating. You're some species, and like there's a dry season and a wet season. Why do you want to mate during the dry season? So you're giving birth during the wet season. Your kids are much more likely to survive. This is an amazing invention, because if instead you're ovulating any old time, and as a result, you're giving birth to kids in the middle of the dry season, you're giving birth to them at the worst possible thing, there's much less likelihood of your offspring surviving. Suddenly, seasonal mating is this great invention, and it is built around one of our macro amplifying modular juggling things, so that the way by which your kidney used to know if it's dry out, retained water, now your fallopian tubes, your ovaries, whatever, know that if it's dry out, start the works going there for mating, because you'll then have a kid during their wet season. And that's a possible pathway by which you get speciation, different times of year when there's mating. Another possible if-then clause. In this case, this is straight out of Monday's lecture. Whatever it is working in our system here where you can recognize this is a sibling, that's great. And okay, let's reverse these. This is my sibling. You recognize that, and that activates some transcription factor, which, as a result, you've got a whole bunch of pathways activated in your brain where you say, mate with them. Yuck, I love them to bits, but I love them like a sibling because they are a sibling. I'm not going to mate with them. You get mechanistic bases of incest avoidance. And thanks to something being juggled, and suddenly a new combination of different parts of different genes by juggling exons, or juggling which exons are under the control of which promoters, suddenly you've got an additional thing. Ooh, if I get the environmental signal that activates this is my sibling transcription factor, and suddenly it's now also turning on the gene for cooperation. The gene. We know it's turning on a network, modulated by environment in different ways, blah, blah, blah. But suddenly, what you have is like kin selection. Suddenly, you've got a great mechanism for it. If I'm getting all these signals that this is my sibling, be more cooperative with them. And we can invent some completely hypothetical pathway between like seeing or smelling your sibling and what that does in the brain and turns on these genes that have something or other to do with being nice and cooperative. Who knows? You see an if-then clause being switched here. Another example. So, progesterone. Progesterone is a steroid hormone, so it works in this sort of way. It has a modular receptor with a hormone binding domain and a DNA binding domain binding to a promoter, all of that. And progesterone is progestational. What it does is mediate all sorts of aspects of female reproduction. Most sort of proximally, it causes the wall of the uterus to thicken. So you've got an if-then clause built into you, which is if there's lots of progesterone around because it's a particular time of cycle or whatever the rule is for how environment is activating this hormone, you got this rule, you start thickening your uterine walls in preparation in case during ovulation an egg is fertilized so it could implant. And if that doesn't happen, go through the period and slough off those cells there. Well, okay, so we've got this if-then clause. If something has just caused a whole lot of progesterone to be secreted, maybe some sort of endocrine signal showing that you are in the follicular stage, but thicken your uterine walls. And then we've got one of our macro-amplifying mutational things happen, and suddenly progesterone is part of a different network. Progesterone, in addition to be part of this if-then clause network, suppose what it now does is it does something in the brain where it decreases anxiety. Okay, that's cool. Is that actually the case? Yeah, progesterone has anti-anxiety effects, and there's lots of evidence, we will eventually get to this, that some contributions to perimenstrual mood changes is because it's coming right after a big drop in progesterone levels. There's some evidence that post-parturition depression is in part driven by the huge drop in progesterone at the time. Progesterone has some anti-anxiety effects in the brain, and people went and figured it out. This is this whole neurotransmitter system. This is minutia. Ignore this if you're like barely holding off. But just for people who care about this, this is a receptor for the neurotransmitter GABA. So it binds GABA and it does something or other to the neuron. Who cares what? And what people then discovered was on the side was a tiny pocket here that binds progesterone. That somehow evolved along the way, and we stumbled, and suddenly now GABA is a very inhibitory neurotransmitter. It calms down the nervous system. GABA has anti-anxiety effects, benzodiazepines, tranquilizers work through the GABA system. But now, thanks to one of these little mutational things that somewhere along the way, something happened in the gene coding for the GABA receptor, and suddenly there was a part of it there, so now the protein can bind progesterone. We've just switched our if-then clause. If there's lots of progesterone around, not only do you thicken your uterine walls and keep doing all sorts of stuff like that during pregnancy, but now you have this hormone having anti-anxiety effects. You've just mixed and matched and juggled things, and out comes completely new if-then clauses. And a way to translate our if-then clauses into what we had 2 days ago, what the change in if-then clause is a change in a regulatory pathway. The evolution of regulation is more important than the evolution of the genes themselves. And here we see right off the bat a mechanism by which you could speciate by inventing seasonal mating or some such thing like that, or you can get dramatic changes. And you can even see this in a sort of experimental way. And this is work actually that my lab did some years ago. So we get back to steroid hormone receptors. So we've got the estrogen receptor, which has a hormone binding domain, which, watch this, using this magnet, a hormone which binds estrogen. And once it binds estrogen, there's the DNA binding domain, which has a conformational change and reveals a promoter, so it could plug into this. Meanwhile, 4 counties over, we've got a different steroid hormone, glucocorticoids. Glucocorticoids, they're secreted during stress. So you've got your glucocorticoid hormone binding domain and the glucocorticoid receptor. It goes in and erases the arrow in the process, I see. And it's got its DNA binding domain, which binds to a completely different promoter. And just to simplify things, like you cannot believe, activating estrogen, activating expression for genes related to making estrogen, it's not actually estrogen, it's the enzymes that make estrogen, has good outcomes, and you are happy. Activate the glucocorticoid pathway, and you get bad outcomes. All sorts of realms. Glucocorticoids increase anxiety, risk of depression. Estrogen tends to have the opposite effect. Estrogen enhances all sorts of aspects of learning and memory. Glucocorticoids do the opposite in a lot of realms, but let's focus in on one particular realm. So, estrogen causes you to make a lot more of these very helpful things called antioxidants in the brain, telling you that all sorts of genes related to antioxidants are under the control of estrogen by way of an estrogen promoter, an estrogen responsive promoter upstream. And meanwhile, glucocorticoids in the brain decrease transcription of antioxidants. When do you want to have a lot of antioxidants in your brain? When you've just had some sort of thing like a stroke, which generates a lot of oxygen radicals, and you really want to have a lot of antioxidants on board. And what you see is estrogen is protective against oxidative damage after a stroke. Glucocorticoids make things worse because they suppress the expression of genes that make something that's going to defend you against these oxygen radicals, antioxidants. So estrogen is just what you want to have in your bloodstream after you've had a stroke. Glucocorticoids are the last thing on earth that you want. And there's a problem here with this in that a stroke is pretty damn stressful. You get somebody who's just had a stroke or cardiac arrest or all sorts of different neurological disasters, and those trigger among the highest levels of glucocorticoid stress hormones you ever have in your body. Jesus, worst possible time you're secreting a hormone which is screwing up our defenses against oxygen radicals. So what my lab did was to try to accelerate evolution in that we made a chimeric, an artificial steroid hormone receptor by fumfering with a gene, mixing and matching exactly along these lines, so that we now had a novel steroid hormone receptor which binds glucocorticoids because it has the hormone binding domain from the glucocorticoid receptor and the DNA binding domain from the estrogen receptor. And what was the hope that this novel receptor gene that we had just invented would make for a receptor which, during times of stress, you would activate protective estrogen genes. Times of stress, symbolized here by all of these glucocorticoid molecules flat, we have just engineered the brain's stress response to be helpful instead of damaging. And what we were able to show was this protected more neurons and more antioxidants, and it also improved learning and memory in these animals during stress. So because instead of glucocorticoids screwing up cognition, you were getting estrogenic protection for free, all of that. This is an artificial version of a macroevolutionary change. You mix and match different pieces, and you suddenly have a very different network. This is now a network where stress makes you less anxious instead of more, where stress makes you more likely to have a sort of intact memory than others are just taking advantage of this. So a very artificial version here of accelerated evolution. What winds up being the downside of this? So we've just seen all these ways which have one little mutation in a transcription factor gene or in a promoter for this, and you get huge consequences, and it changes entire networks, and you invent completely new networks under the control of this transcription factor. What are the odds that this is going to produce something helpful, something that's actually advantageous? What you've just done is, what have you just done? You've just like mixed and matched like the opening sentences from 14 different novels so that they're now like, what are the odds that something interesting and helpful and coherent is going to come out of this? Very unlikely, because as we saw the other day, go have a point mutation, a simple one in one single gene, and you're changing the likelihood of ADHD or where you are on the autism spectrum, or where you, like just one little classical mutation. Now you're changing entire networks that weren't there before. What are the odds that that's going to turn out to be advantageous? Really, really unlikely. In other words, what you're seeing is most of the time, these mechanisms for macroevolutionary change are a disaster and get selected against. And thus, things remain the same. Things remain static. This is a mechanism for long periods of stasis in certain properties because, like, roll the dice and get some random mutational thing happening, and the odds of you now suddenly getting the most helpful network on Earth anyone ever thought of is really, really slim. Most of the time, these are disasters. Most of the time, they're stasis. When do you suddenly get these rapid changes that are beneficial? Something that like population biologists call selective bottlenecks. Something has gone on. There's a volcano that's erupted. There's an asteroid. There's a change in the temperature of the water. There, something happens which is a disaster and kills 99% of the individuals in a population. And it turns out the only one, or the 1% that survive, have recently stumbled into a completely novel network where, what do you know, what are the odds that this happened to be essential for surviving this new circumstance? You get a selective bottleneck, and only a small subset survive because of something genetically unique in them, and they're now all descendants of that. And the best evidence is humans went through an evolutionary bottleneck, a selective bottleneck. Oh, I don't remember the numbers, 200,000 years ago or something, where I don't remember this number either, but like we were down to something like 5,000 humans go after some selective bottleneck, and all of us are the descendants of that, and we came within an inch of going extinct. Those are the sorts of circumstances where what is normally disastrous and thus maintaining stasis instead can suddenly luck out under the extraordinary circumstances, meaning every now and then. Okay, so we now see these molecular mechanisms from the other day for gradualism, for dramatic jumps, all of that. What does it actually look like out in the world there? Do you ever actually see evidence for evolution happening before our eyes? And when we do, is it gradualist or are there dramatic changes? And by definition, It is very hard to see gradual evolutionary changes because you don't want to spend, 400,000 years trying to do your doctoral thesis, and you got to see if your like ferns are going to evolve into a new species. Gradualism, by definition, is slow. But there's all sorts of examples of rapid evolutionary change, rapid not only from the standpoint of these snail paleontologists, but even rapid in our senses over the course of a few generations. One example, the famed Siberian foxes, which is one of the coolest stories out there. There was this Russian geneticist, Leovev, in 1960, did this study where he had sort of like a whole farm of silver foxes, and people did a lot of that, made a lot of these for farming, something horrible, killing them for that. And he decided to do an experiment. He took one of these populations of captive silver foxes and he controlled their mating. He did artificial selection on them. He would simply go up and see you walk up to this particular fox, and does it run away from you because you're some scary human? Let's find the few who don't seem as skittish around humans. Let's breed them. And then let's breed of their offspring, the ones who are least skittish around humans. Let's selectively breed them. And what they showed was over the course of 35 generations, you turned these silver fox things into these like little domesticated, adorable foxes where they loved licking your face and jumping up into your arms, and they would wag their tails. Foxes do not wag their tails, but over the course of the selection, they started wagging their tails. This was basically artificially domesticating a species over 35 generations. Totally cool. And those pictures are so cute of those domesticated silver foxes. Something else interesting happened in the course of this. So you're selecting these foxes just for behavioral traits. They're less skittish around humans. Eventually, you're selecting for ones that are particularly curious, whatever, more affiliative and weird. You're selecting for behavioral traits of them becoming domesticated pets. Interestingly, there were other changes in these foxes, which was over the course of these generations of being bred for a behavioral trait, what started to happen was they got different musculature in their tails, so they could wag their tails. Their snouts started shortening. Their ears got little and rounded. Their foreheads became more roundy and stuff. What did they do? They were becoming cuter. They were becoming cuter. They were developing cute attributes, which made them even more adorable as a domestic species, and even more likely that you want to invest your grant dollars in getting kibble for your domesticated fox. Their appearance changed. And what people began to realize was the mechanism for this is they neotenized. They became more childlike. They became more infantile in their behavior and their appearance, which is exactly what went on with the domestication of dogs. Dogs are baby wolves, what they were bred for in this co-selective process by which dogs and us domesticated each other. But what went on there was, in addition to selecting for behavioral traits, what you are selecting your dog to be is, no matter how old it acts like a little baby wolf. It's super affiliative, and it's non-aggressive, and it thinks you're mom, and thus it is your faithful dog at the side there who will sit by your grave for decades after it. What domestication is about in species after species is breeding for neotenous, infantile traits, infantile behaviors, infantile appearance, short little muscles and cute little ears and all of that, which we'll hear about in ethology lectures, and a less developed brain. Dogs have smaller brains than wolves. They're not as smart as wolves. Part of what went into making a dog into a dog was you had to make them act like baby wolves who were not very sharp. And thus, what was happening in what has now been termed the domestication hypothesis is in the process of 35 generations of breeding these silver foxes into them acting like domesticated pets. They began to look like domesticated pets as well. They began to look like babies, and before you know it, they're just so cute. You want to scream, you want to pinch their cheeks, all that sort of thing. And just as foreshadowing, the dog example of dog domestication, making dogs into essentially baby wolves behaviorally, when you sit there, you, as the studies show female, you have your beloved dog and you stare into each other's eyes, you both secrete oxytocin, a hormone having to do with mother-infant bonding. And as shown in the study, which we'll eventually look at in more detail, if you boost up the oxytocin levels in either the human or the dog, they look in each other's eyes longer. This whole domestication process is domesticating wild animals into looking, acting, feeling, thinking like babies in a way that makes you look, act, think like their mother, regardless of what sex you have. So the most important thing about this is this was done in 35 generations. Incredibly rapid evolution for these traits. So what we will pick up on after this next week is more examples of super rapid evolution in animal species in us as well. And then looking conversely at examples of gradualist evolution that has occurred in real time, where we can see it. And then how do we go about reconciling these two different versions, these two different realms by which you have mutational changes, and thus a protein is working 2.5% better or worse than it used to, versus things like this, where you change entire networks and you generate new species overnight.